Hi everyone, I am Dr. C.A. Sulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with cholesterol and beta cells. In today's session, we will start with oxidized metabolites. Then we have formation of bile acids, including all steps. Then we have functions and secretion of biles. And lastly, we have a new chapter called MCFA, PUFA, prostaglandins, and compound lipids. Good morning, everyone. Let us begin the topic. Oxidized metabolites. In contrast to their parent molecule, cholesterol, the oxidized metabolites are able to cross blood-brain barrier. In the concentration-driven flux of 24-hydroxycholesterol from the brain to circulation, which is of major importance for elimination of excess cholesterol from the brain. On the other hand, the opposite flux of 27-hydroxycholesterol from the circulation to the brain may regulate a number of key enzymes within the brain. In vitro experiments suggest that the balance between the level of these two molecules may be important for the generation of beta amyloid peptides. In primary cultures of rat procampal cells, 27-OHC is able to suppress expression of the activity regulated cholesterol associated protein, that is ARC, a protein important in memory consolidation, which is reduced in patients with Alzheimer's disease. Bile acids are synthesized in the liver from cholesterol. They contain 24 carbon atoms. All of them have an alpha oriented, that is projecting below the plane of ring, hydroxyl group at position 7. The reactions for synthesis of bile acids are summarized below. First, there is cholesterol hydroxylated at 3, 7, 12 positions. Then the removal of 3 carbon unit to make it 24C. Then conjugation with glycine. Then secretion of intestine. And lastly, in the intestine, conjugation and removal of hydroxy group. Part 1, Hydroxylation reactions. The first and rate limiting step in the introduction of this hydroxyl group by the enzyme are 7-alpha hydroxylase. It is a microsomal enzyme. Then the beta-oriented OH group of C3 is converted to alpha type by an isomerase. The third OH group is added at the 12 carbon in the case of folic acid. Here you can see cholesterol is converted into 7-hydroxycholesterol with the help of enzyme 7-alpha hydroxylase, which is a classical pathway. Chenodeoxycholic acid, another primary bile acid, has 2 hydroxyl groups at position 3 and 7. Ring B is reduced in all cases. The side chain is first hydroxylated at 26C and then oxidized to COOH group. This is followed by cleavage at 24C with removal of propionic acid, that is 3 carbon unit. In the diagram you can see that in the upper part there is cholic acid, that is 3,7,12 trihydroxycholanic acid, whereas in the lower half there you have chenodeoxycholic acid, which is 3,7-dihydroxycholanic acid. The third part is formation of bile salts. The primary bile acids are now conjugated with either glycine or taurine to form bile acids. They are glycocholic acid, taurocholic acid, glycochenodeoxycholic acid, and taurochenodeoxycholic acid. The major conjugated bile acid is glycocholic acid. Conjugation adds more polar groups and increases the efficiency of bile acids as surfactants. The conjugated bile acids and excreted through the bile. In the bile, they exist as bile salts, that is sodium or potassium salts of conjugated bile acids. In the diagram you can see glycocholic and taurochenodeoxycholic acid is formed. Cholesterol has got two paths. First is plus 3 OH group, which is converted into cholesterol 3-12-7-12 triol, then cholic acid, then cholyl CoA, then glycocholic acid. On the other hand, it has plus 2 OH groups, which includes cholesterol 3,7-diol, then chenodeoxycholic acid, then chenodeoxycholyl CoA, and finally taurochenodeoxycholic acid. Next part is secondary bile acids or bile salts. Primary bile acids are acted upon by intestinal bacteria which result in deconjugation. The deconjugated bile acids are then partly converted into secondary bile acids by removal of alpha hydroxyl group at position 7. Cholic acid is thus converted to deoxycholic acid and chenodeoxycholic acid is converted to lithocholic acid. Coming to the functions of bile or salts. They facilitate the digestion of lipids. They can form molecular aggregates called micelle which bring about absorption of lipids. Bile salt micelle is also an important role in keeping the cholesterol in solution. During the last 2 decades, it has been discovered that bile acids are regulatory molecules. Bile acids have been discovered to activate specific nuclear receptors, G protein coupled receptors EGR5, and sex signaling pathways in the cell, in the liver and gastrointestinal tract. The activation of nuclear receptors and cell signaling pathways alter the expression of numerous genes encoding enzymes or proteins involved in the regulation of bile acid, cholesterol, fatty acids, lipoproteins synthesis, and metabolism and transport. Bile acids appear to function as a nutrient signaling pathway molecules, primarily during the feed or fast cycle, as the efflux of these molecules returning to intestines to liver following a meal. Bile acid controlled signaling pathways are promising novel drug agent targets to treat common metabolic diseases such as obesity, type 2 diabetes mellitus, hyperlipidemia, and atherosclerosis. Coming to enterohepatic circulation of bile salts. Of the total bile salts reaching the intestine, that is 15 to 30 gram per day, only a very small fraction, that is about 300 to 500 milligram per day, is excreted through feces. The rest is absorbed into ileum, reaches the liver and re-excreted through the bile. In this diagram, we can see enterohepatic circulation of bile salts. First, they are secreted by the liver, then they pass through common bile duct, then in the duodenum, then in ileum. In the ileum, they are reabsorbed by the capillary bed and and once again sent back to liver for enterohepatic circulation. Coming to the next point, bile. It is a chief secretion of liver, the largest gland in the body. Daily volume of secretion is about 500 ml. Thus, secreted bile is stored in the gallbladder and released on demand. The pH of bile in hepatic duct is 7.8, whereas that in gallbladder is 7.4. The enzyme present in the bile is alkaline phosphatase. Coming to secretion of bile. Cholerectics are substances which stimulate the secretion of bile by the liver, whereas cholagogues stimulate the release of bile from the gallbladder. The most important cholerectics are bile salts, hormone secretin, and vagal stimulation. Cholecystokinin is the most powerful cholagogue. Coming to functions of bile. The alkaline pH of bile serves to neutralize the acidity of the gastric juice. The bile salts are efficient surfactants and detergents. Bile is the only route of excretion for bilirubin, which is the end product of heme catabolism. It is it serves to excrete cholesterol, thus regulating the body cholesterol pool. Bile serves as a medium of excretion for several drugs, such as which are detoxified by the liver. Coming to solubilization of cholesterol in bile. In the relative concentration of cholesterol in the bile favors the precipitation and resultant stone formation. Hence, it is referred to as erythrogenic bile. Bile salts and phospholipids in the bile play a significant role in keeping the cholesterol in solution by forming mixed micelle. Infection of the gallbladder may produce to stone formation. Dehydration may also lead to precipitation of cholesterol crystals. Coming to cholesterol and cardiac diseases. The level of cholesterol in the blood is related to the development of atherosclerosis. Abnormality of cholesterol metabolism may lead to cardiovascular accidents and heart attacks. Next, coming to next chapter, medium chain fatty acids, polyunsaturated fatty acids, prostaglandins and compound lipids. Coming to introduction, fatty acids having carbon atoms 4 to 6 are called short chain fatty acids, 8 to 14 are called medium chain fatty acids, 16 to 18 are called long chain fatty acids, and 20 or more are called very long chain fatty acids. SCFA, that is short chain fatty acids, contain butyric acid and caproic acid, are seen in butter and ghee. Medium chain fatty acid, that is capric acid, lauric acid and myristic acid, are present in coconut oil and human milk. Normal skin has a very thin layer of sebum secretion with MCT, that is medium chain triglyceride, which containing lauric acid, which prevents bacterial entry into the body. Coming to digestion of medium chain fatty acids. Digestion and metabolism of SCFAs and MCFAs are drastically different from those of LCFAs. Short or medium chain fatty acids contain triglycerides, that is SCT and MCT. They do not require prolonged digestion. Pancreatic lipase and bile salts are not required. MCT specific lipase catalyzes the complete hydrolysis of SCT or MCT into glycerol and short or medium chain fatty acids. These free MCFAs diffuse directly into the portal circulation. SCFA and MCFA are preferentially oxidized by peripheral cells, and so they do not deposit in adipose tissues. Here ends our session. Kindly like, share and subscribe the channel. Thank you.